And what I want to talk about, what I started talking about last time was promoters. And so I want to finish up, clarify things on promoters. So I'll remind you, first of all, a promoter is a sequence near where transcription starts. And why does transcription start anywhere? Transcription starts because the cell has to make an RNA, and cells are making RNA so they can make proteins. That's why they're making RNAs. They're making RNAs so they can make proteins. Most commonly, we think about them making the messenger RNA necessary for coding for a protein. But transcription also includes the synthesis of ribosomal RNA, which is used in the synthesis of a protein. It's also used for the transfer RNA, which is also used for the synthesis of a protein. But most commonly, we think about it with respect to the messenger RNA. So what you see on the screen was what I started to talk about yesterday, which was the interesting sequences that are about 10 bases ahead of. And by the way, when we say ahead of with respect to sequences, we usually say upstream. Okay? So upstream of the start site, which means in the 5' direction, which on this image is to the left, okay, we see this sequence that's very commonly found there called the Pribnow box. And as I noted yesterday, AT base pairs are very easy to take apart. And this provides an entry point for the RNA polymerase. The RNA polymerase is recognizing and binding to the sequences that you see on the screen. And the efficiency with which it binds to any of these sequences is related to the actual sequence. So as I noted yesterday, the closer a sequence is to this TATAAT, the more likely RNA polymerase will bind there and transcription will start. So I talked about strength of promoters. A strong promoter is a promoter where RNA polymerase binds a lot and makes a lot of messenger RNA. Yes, question? Why is it 10 bases ahead? It turns out that 10 bases is about 1 turn of the DNA. And so it appears that it's binding and that the part of the polymerase reaches out about that distance. So the distance from where the actual polymerization part of the RNA polymerase is compared to the part that's binding the DNA is about that 10 bases. Okay? Okay. You'll notice there's also a blue sequence labeled up here, and this blue sequence is called the -35 sequence. And you'll see that every one of these has a -35, and the -35 isn't quite as conserved, as we say in the sequence biz, as the Pribnow box sequence is, but it's fairly commonly through there. Okay? And again, we think about the more related this sequence is to this idealized sequence at the bottom, again, the stronger the promoter is. Now, I'm going to talk about a protein. By the way, RNA polymerase is a multi-subunit protein. Multi-subunit protein. And so some of these subunits have specific functions that we're going to talk about. So some of the specific subunits of RNA polymerase have specific functions that we'll talk about. And one of those functions I'll talk about is the recognizing the promoter. Okay? If we look at the sequence of events that happens in transcription, we can break it down into initiation, elongation, and termination. So it's the initiation phase that I want to talk about with respect to the recognition of the promoter. Yes, question? Oh, is it doing that thing again? Okay. Let's see. How about... Did I get it? That got it. Yeah, thanks for telling me. Old guys don't hear those things, so... Okay. I give you enough of a headache as it is, so I don't need to have the system making it even worse, right? So, all right. So initiation. In order for transcription to start, the polymerase has to find where it's supposed to go. So it's got to find the promoter. One of the subunits of the RNA polymerase is a factor called sigma. And you can see it right there on the bottom of this thing that says RNA Pol. Okay? Sigma's job is to recognize and bind to the promoter. since the sigma is linked to the RNA polymerase, it's actually carrying the RNA polymerase to a promoter sequence. Sigma will have affinity, meaning it likes to bind to certain sequences. And it turns out that there are actually different sigma factors that bind to slightly different promoters. Okay? Different sigma factors that bind to slightly different promoters. For our purposes, we're going to think pretty much of the TAT-AAT minus 10 sequence. But there are other sigma factors that will bind to other sequences related to that. Okay? Well, in the initiation phase, the binding has to happen, and you can see in the binding that there's unwinding of the DNA. So as I mentioned yesterday, I got the hiccups. As I mentioned yesterday, the strands have to be pulled apart, and RNA polymerase has to get in between those strands to do its thing. Okay? This is what we call, in fact, you can see it right here, a transcription bubble. It looks like a bubble. And so what's going to happen in the process of transcription is going to move to the right, okay? And that bubble is going to move with it. So as the transcription proceeds, that bubble is going to continue moving down, in this case, to the right. Okay? So you can see here that the transcription process has started. You see this little thing that says purine, Pu. That doesn't mean it stinks. It just means purine. All right? With 3 phosphates attached. And we see that the process has started. You see these N's here. N means any nucleotide, which is whatever it happens to be recognizing on the bottom strand. The strands have names. Okay? The strands have names. The strand that's being copied, which in this case is the bottom strand, and by the way, notice that the strand at the bottom is running 3' to 5', but the polymerase is running 5'. The polymerase is making RNA in the 5' to 3' direction because they're anti-parallel. Okay? The bottom strand is the one being copied, and the bottom strand is called the template strand. The top strand is not being copied, and it's called the coding strand. And the reason it's called the coding strand is because it's complementary to the bottom strand. It has the same sequence as the RNA does, except for the RNA has U instead of T. So the top strand, in this case, the one that's not being copied is the coding strand. The one that is being copied is the template strand. Okay. Well, this process of initiation starts up here, and it ends down here at about the end of putting together about 10 nucleotides. The first 1 to 10 nucleotides, we call that part of the process initiation, and then after that, we move into another phase. And we move to another phase because what happens is you'll see that at this point, sigma is released. Sigma's only job in the process is to help the RNA polymerase to recognize the promoter. Once the process has started, sigma is no longer needed, it's released, and the polymerase continues on without it. Okay, at this point we've finished initiation. Well, elongation is the easiest of the phases because it simply means that RNA polymerase copies that template strand using the base pairing rules that you already know. We encounter the last phase of the transcription process, which is the termination phase. And the termination phase is kind of interesting, and it can occur by 2 different mechanisms in E. coli. So we're still talking only about E. coli here. All right? 2 different mechanisms. All right? The first one of these is called the factor-independent method. It's probably the more difficult of the 2 to understand, but it's not too bad. Okay? I'd like you to think back to the last figure I showed you right here. RNA polymerase is moving along. It's copying the DNA. And you'll notice that what's happening is the RNA is sort of leaking out the very bottom of this thing. This RNA will get longer and longer and longer the farther to the right that the RNA polymerase goes. Okay? So I want you to imagine this tail of this polymerase, which I like to think of as sort of flopping around there. This tail is sort of flopping out the end of this, and it's getting longer and longer and longer as the polymerase is progressing. Now that's important because when we get to a certain sequence that the polymerase copies, if it finds the sequence I'm showing you on the screen, something interesting happens. Okay? So the polymerase is moving along. We have this long tail that's hanging off. And here are some sequences that the polymerase has actually copied. You can see there it's RNA because it has U's in it. Well, you'll notice that this sequence, the polymerase makes it, and then it continues to make it, and then, whoa, this is complementary all the way down there. This strand is pairing with itself. You saw things pair with themselves before when I showed you the tRNAs or when I showed you the ribosomal RNAs. But here is a messenger RNA sequence that's pairing with itself. Now, this is actually in the DNA for this gene. Okay? It's in the DNA for this gene. This thing, this duplex, because this sequence is immediately next to this, will form all by itself. Nothing else is needed for it to form. Those base pairs will form all by themselves. And I like to think of this thing forming as like a jack on a car. All right? If you wanted to lift the car up, you would jack the car up, and the rear end of the car would get lifted, and you can change the tire, right? Well, this thing, when it assembles, kicks the butt end of the RNA polymerase up. Remember, the RNA polymerase is sitting there in that bubble, and this thing has formed right underneath the RNA polymerase. It lifts the RNA polymerase up, and when that happens, the whole bubble complex falls apart. The whole bubble complex falls apart. What we have just done is we have just terminated the process of transcription. And it got terminated because this sequence literally kicked the RNA polymerase off of the DNA. Can you envision that? Could you describe that to me on an exam? Okay, can I do it again? Yes. Okay, so the RNA polymerase is moving along. It's got this long tail flopping off of the polymerase. We have the RNA polymerase is on the DNA, and it's in that bubble. It encounters a sequence in the DNA that this is complementary to. So when it makes this sequence goes boing and forms a duplex like you see here, which lifts, it actually forms underneath the RNA polymerase, and it lifts the polymerase up off of the DNA. That causes transcription to stop and everything to fall apart. So it's called factor independent because it doesn't require any external factors in order to stop transcription. The next method I will show you for termination does involve other factors, but they're actually easier to understand, I think. Other questions about this? Yeah? So this... Sure. Okay. So what's happened is, and this figure doesn't show it very well, but the RNA polymerase has already copied through this sequence, and this sequence, when it's copied, makes an RNA that has this. Okay? So the RNA polymerase would be sitting just ahead of that sequence on the DNA because it would already have gone through this, and then that's when everything falls apart. Okay, no. Okay, so no, you're confused. So she said her question was, once it goes past this first one, right, does it jack up? No, it can't until it gets to the second one because there's the first one right there, and there is the second one right there. So it has to go through both of them before the structure can form. Okay? Has to go through both of them. Okay, this thing is called a hairpin, by the way. You frequently see these in a nucleic acid. When a nucleic acid can fold back on itself and make base pairs within itself, it forms what we call a hairpin. Okay? Sometimes people call it a panhandle also. Okay. Now, the factor-dependent one is another way of terminating transcription. Okay? It's another way of terminating transcription, but instead of using the mechanism I just showed you, it uses a different mechanism. All right? The most common question I get is, well, when does the cell use one and when does the cell use the other? All right? And it depends on the gene, so we're not going to worry about which one is which. You should just know that these are mechanisms of terminating transcription. This method, we can see the bubble right there. We can see the RNA polymerase right there, and we can see this end flopping off of the end of it, this RNA flopping off of the end of the bubble, as you can see right here. The farther this goes to the right, the longer this tail will be, right? This method is kind of cute, all right? This end flopping off of here is a target for a protein called Rho, and that's Rho. It looks like a P, but it's actually a Greek character, Rho, all right? Rho is a protein that's kind of like what you've already seen the beta clamp. Remember the beta clamp made that circle around the DNA? Rho is circular, and Rho does something really cool, all right? So the example I like to give for what Rho does is something that you did probably in high school gym. How many of you had to climb the rope in the gym? Everybody had to do that. But first time I did it, I was terrified, right? And then I kind of liked it after a while. But the first time I did it, it's like, holy crap, you know? I just didn't think I was going to survive that process. So you climb that rope, and you get to the top of the gym, and you're looking down on everybody, right? Well, imagine that you're climbing that rope at the same time as somebody is letting it out. Right? You got a moving target. Now, that really is scary. That's a very scary thing, right? Because you're trying to scramble, and you want to climb faster than the person is letting the rope out if you ever expect to get to the top of the gym, right? Well, that's what's happening right here. The RNA is growing as the Rho factor is moving to the right. So Rho is climbing that rope. The rope being the messenger RNA that's here. Okay? It's climbing that rope. And what happens with Rho is it climbs that rope, and it will climb the rope faster than the rope is being let out. So that Rho will eventually catch up with the RNA polymerase, and then it kicks the butt end of the RNA polymerase up, just like we saw, and everything falls apart. Now, this method is a little bit more variable in where the stop actually occurs. It's a little bit more variable about where the stop actually occurs because this is sort of a race between how fast the polymerase is moving and how fast Rho is moving. Okay? So these won't give exact stop places. They'll be variable in terms of where they will stop. But one of the clues to where it will stop is actually given by the sequence that's being copied in the DNA. It turns out that RNA polymerase, not surprisingly, goes slower through GC sequences. If it hits a stretch of GCs, the polymerase slows down, and when the polymerase slows down, Rho has a better chance to catch up with it. So we will tend to see stop locations for this type of transcription. These stop locations will be around fairly GC-rich regions of the original DNA. Okay. Questions about that? You guys want to slow down? Want me to slow down for a minute? Yes, question? This process is called termination of transcription. So it's the other kind of termination. The first kind was the factor-independent termination. This is the factor-dependent termination. Okay? Okay. Well, in the end, you have a messenger RNA that's made. Now, I keep throwing out this term gene. I assume everybody knows what a gene is, but if you don't know what a gene is, for our purposes, a gene is something that codes for a protein. It's a region of DNA that codes for a protein. Prokaryotic cells are different from eukaryotic cells in the sense that they may have more than one gene on a given messenger RNA. We'll talk about that when I talk about the eukaryotes. Eukaryotes don't have that go on. They only have one gene per messenger RNA. Prokaryotes can have more than one gene there. Okay. So that's the process of termination. And that actually leads me to my next thing. Okay? The more than one gene located there. Bacteria, when we look at bacterial DNAs and we look at the location of genes in bacterial DNAs, we discover that those genes in the bacterial DNA are very tightly packed together. There's very little in terms of non-coding regions between genes in bacterial DNA. Very, very few bases between there that don't code for protein. That's very different from eukaryotic cells. A eukaryotic DNA, the cells that say, the DNA that's in your cells is about 1,000 times longer than the DNA that's found in E. coli. If everything else were equal, you would say, well, we have 1,000 times more genes than what E. coli has. And we don't. We have about 10 times as many genes. E. coli has 3,000 to 5,000 genes. We have 30,000 to 50,000 genes, depending on how you want to count them. Okay? So that tells you there's something different about our genes. Are they that much bigger? No, not really. But there are more spaces. Spaces between genes, and in eukaryotic cells, we also see spaces within genes. And I'll talk about those as well. Well, because bacteria are so efficient in their localization of genes, they're also very efficient in the way that transcription occurs. We're very wasteful the way we do transcription compared to bacteria. Bacteria are amazingly efficient at this process. Okay? Bacteria will have related genes that perform related functions next to each other. Okay? And the organization of these related genes goes into a structure we call an operon. O-P-E-R-O-N. I'm going to define what that means for you. An operon is a collection of genes in bacteria that are all controlled by the same promoter. A collection of genes in bacteria that are all controlled by the same promoter, and the upshot of that is they will all appear on the same messenger RNA. So as I mentioned earlier, a bacterial messenger RNA can have several genes on it. Ours, that is ours humans, eukaryotes, our messenger RNAs will only have one gene on a given messenger RNA. Well, a really good example, and we'll talk about this example in the later in the course as well, is the Lac Operon. Okay? The Lac Operon. The Lac Operon is, let's see, is actually better shown here. Let me just start here to show you the Lac Operon. Okay? The Lac Operon consists of 3 genes called LAC-Z, LAC-Y, and LAC-A. Each of these genes has a function, and each of these genes plays a role in the bacteria's ability to metabolize the sugar lactose. That's why it's called the Lac Operon. Each of these plays a role in the ability of the bacterium to metabolize lactose. Now, bacteria are very efficient, as I said. Bacteria spend a lot of their time running around looking for food, and as soon as they find food, they start gobbling the food and they start dividing like crazy. And when they don't have food, they're not like us. They don't have fat reserves that they can live off of where they don't eat for a while. They have to shut everything down or they're going to die. Because if they don't shut things down, they're going to burn up all their energy. Now, this actually relates to what this operon's about. Imagine I'm an E. coli, and I'm in the gut of Kevin Ahern. And Kevin Ahern decides he's going to go to the gelato place and have some gelato. Well, gelato, of course, is a dairy product, and dairy products are full of lactose, which, what's that? Is that low lactose? Oh, I picked a bad example. Okay, so I'm going to go have my dryers. You see how it's good to have nutritionists in here, so thank you. All right, so I'm going to go and I'm going to have a milkshake. Is that better? Okay, so I'm going to have a milkshake, right? And I'm going to embarrass myself here. Okay, and milk is full of lactose. Lactose is known as milk sugar, right? So the E. coli that's in my gut, see this coming and going, oh, wow, man, look at all this lactose. This means we've got some energy. We can burn this energy, and we can make copies of ourselves. And so they do that. Okay? And then, after I've had that for a while, and I said, man, I'm getting too fat, which I really am. Okay? It's time to drop some weight. I'm going to really cut back on what I'm eating, and I'm not going to have any milkshakes, and I'm going to go eat gelato. I'm not going to have any dairy products that have much lactose in them. Okay? What's going to happen then is these bacteria in my gut are looking for lactose, but there's none there. Does it make sense for them to be making proteins that break down lactose if there's no lactose there? Of course not. That's wasting energy. So the cells, the bacterial cells, have an ability to sense if lactose is present. And if it's present, they turn on the synthesis of these genes. These genes will then break down lactose, and everybody's happy. When these cells run out of lactose, they said, okay, quit making these genes because we don't need to break down lactose because there's no lactose to break down. So two very simple situations, lactose, no lactose, right? The operon that I'm going to describe to you, or the control of this operon that I'm going to describe to you, allows the cell to recognize and respond accordingly. Okay? Now, for our purpose, I'm only going to talk about one of the genes. It's called the Lac-Z gene. They all play a role in the metabolism, but the Z is the easiest one to understand. Z breaks down lactose into the two sugars that make it up. It breaks it down into galactose and glucose. That's what it does. Okay? It breaks down lactose into galactose and glucose, and then we can burn those up, or the bacterium can burn those up. We can too. All right. Looking at what's up here, all right, this shows how this control is actually exerted by the proteins in the cell. So before I tell you this, I have to tell you something that I haven't described before. And that's something called a transcription factor. A transcription factor. A transcription factor is a protein that binds to a specific sequence in DNA and affects transcription. That effect can be positive, that effect can be negative. But a transcription factor is recognizing and binding to a specific sequence in DNA and affecting transcription. Okay? There are two transcription factors in this operon that we're interested in. Okay? One of these is something called the Lac repressor. That's the most important one, the Lac repressor. The Lac repressor is a protein. It finds a sequence in the promoter region of the operon, and it binds to it. You can see the Lac repressor right here. It actually forms a tetramer. There's 4 of them that come together. And you can see that it has bound this region up here called O, which is actually where the promoter is. The repressor, when there's no lactose present, will bind to that promoter region. And the reason it's called an O is because this promoter region is called an operator, for what it's worth. It's called an operator, like telephones used to have. Okay? The repressor binds to this promoter region called the operator, and it sits there and it does nothing. But because it's sitting there and covering up the promoter region, the RNA polymerase can't bind. And if the RNA polymerase can't bind, transcription can't occur. The repressor has deserved the name that we give it. It has repressed transcription. It has stopped transcription from occurring. Okay? This is what happens when there's no lactose. Well, what happens if there is some lactose? Well, if there's some lactose, a little bit of it will leak into the cell. And when it leaks into the cell, lactose will bind to the repressor. The repressor actually has a binding site for lactose. And I'm actually lying to you just a little bit. It's a molecule that's like lactose, but for our purposes, we'll say it's lactose. Okay? I like to keep it simple. Lactose binds the repressor, and when it binds the repressor, something interesting happens. The repressor is no longer able to bind DNA at that point. Well, if the repressor can't bind the promoter and cover up the promoter, RNA polymerase now has access to it. RNA polymerase can bind the promoter and start transcription of these genes. Now, this is a very simple but a very cool system. When lactose is present, transcription is allowed, and these genes that break down lactose are made. And when lactose is absent, transcription is turned off because the repressor is activated. It binds and covers it up and stops transcription from occurring. Very simple. Okay? And simplicity is good when it comes to cells. Cells have enough complicated things in them already. But very simple switch. On or off, depending upon whether lactose is absent or present. Does that make sense? Okay. There's one other protein I want to talk about that's relevant here. And it's called a protein called CAP. C-A-P. Okay? It has a longer name, but we're not going to worry about it. Okay? Here's that region of DNA that I was talking about before. You can see the LAC-Z gene over here. You can see the operator, which is where that repressor would bind if it was present. You can see the RNA polymerase has now bound because there was no repressor bound. And you can see this protein called CAP that's there. And you'll see CAP has something next to it that says dash CAMP. CAMP is a special molecule inside of cells that conveys signals. We'll talk more about it in the term later. It's a modified form of the nucleotide AMP. So you know ATP, ADP, AMP. This is a modified form of AMP. It's very commonly used inside of cells to communicate signals. One of the signals it's communicating here is, I'm very low in energy. Cell is very low in energy. Okay? When the cell's low in energy, it makes cyclic AMP, CAMP. And CAMP is capable of binding to this protein called CAP. And CAP is a transcription factor as well. CAP binds over here And cap does something very important. Cap actually helps the RNA polymerase to bind. Cap, when it's bound to DNA, helps the RNA polymerase to bind. And the only time that cap will bind to DNA is when cap is bound to cyclic AMP. Only when cyclic AMP is present will cap be bound here. Now, this might seem a little screwy, but it's not. Imagine the cell is low in energy. It makes cyclic AMP. Cyclic AMP binds to cap. Cap binds here and helps the RNA polymerase. You might say, well, we've already got a promoter there. Why do we need cap? The reason is the RNA, the promoter that's there for the Lac Operon is not a very good promoter. It's a weak promoter, which means that the RNA polymerase is going to bind some at the time, and it's going to make a little bit of RNA, but it's not going to make a lot of RNA. However, when the cell is starving and lactose is present, you'll want that cell to be cranking out the genes that break down lactose. So cap binds and helps the RNA polymerase to now get in there more frequently and turn what was a weak promoter into a strong promoter. You get a lot of messenger RNA made when cyclic AMP is present. Okay? Questions about that? Let's imagine we have the repressor. We have no lactose, and the cell is running out of energy. That means you're going to have cyclic AMP binding to cap, cap binding to this sequence, and since there's no lactose, the repressor is going to bind over here. Who's going to win? Is cap going to stimulate transcription, or is the repressor going to win and turn off transcription? What do you think? How many think the repressor wins? How many think cap wins? You guys are good. The repressor wins. Okay? And it makes sense that the repressor would win because, again, you don't want transcription occurring if there's no lactose present, which is what you have. Okay. Now, when we talk about eukaryotic cells, you will thank me for not going into the detail. This might seem a little complicated to you. There are dozens of proteins that can bind to promoters in eukaryotic cells. And they can bind to promoters that are very close to the start site. They can bind to promoters that are thousands of bases away and have very, very strange effects on them. And we're not going to talk hardly about them at all. Everybody's falling asleep here, I can see. Okay. All right. I'll talk very briefly about one other operon, just because it's kind of interesting. But I'm not going to expect you to go into detail on this mechanism. All right? This mechanism is a cool mechanism, though. It's called attenuation. A-T-T-E-N-U-A-T-I-O-N. Attenuation. Okay? This method that this control mechanism for transcription that I'm going to talk to you about involves the tryptophan operon in E. coli. Okay? This is an operon that the E. coli cell uses to make tryptophan. So E. coli can make their own tryptophan. And we've got a similar situation like we had with respect to lactose. If the cell is needing tryptophan, it wants to turn on the genes to make tryptophan. And if the cell has plenty of tryptophan, it doesn't want to make those genes and waste that energy because it doesn't need any more tryptophan. With me? Okay. Well, this turns out for tryptophan to be a pretty important consideration because the tryptophan operon has 10 genes in it. That's a lot of messenger RNA. It takes a lot of energy to make a messenger RNA. It takes a lot of energy to make protein. And so the cell really wants to be able to control this properly according to need. It doesn't want to waste energy, and there's a lot of energy that can be wasted if this system doesn't work properly. Okay? This sequence, okay, and it's very simply shown here, is a very interesting mechanism. This sequence does not control whether transcription starts. It controls where transcription stops. So it's a termination control mechanism. Okay? So the tryptophan operon has a promoter that RNA polymerase binds to, and nothing stops it. It always gets started just fine. However, when there are high amounts of tryptophan, that is, the cell has a lot of tryptophan, a terminating sequence stops transcription after only a small messenger RNA is made. Early termination. That happens before the first gene is even complete in terms of the RNA polymerase. This guy falls apart. On the other hand, if tryptophan is low in concentration in the cell, termination does not happen, and the RNA polymerase continues along its merry way through all 10 of those genes. Those genes get made into protein, and those proteins start catalyzing the synthesis of tryptophan. Everybody with me? High, low. Okay? So this mechanism works on stopping transcription, either early or late, depending upon whether tryptophan concentration in the cell is high or low. Now, the mechanism it actually uses, I'm just going to show you one little tiny piece of it, but I'm not going to expect you to memorize the mechanism, because people always get worried about the mechanism. Okay? If you're interested in the mechanism, come see me, I'll tell you. But the one piece I want to show you actually relates to sequences that are in this little piece of messenger RNA. Okay? That little piece of messenger RNA has coding in it for some interesting amino acids. Now, we'll talk about translation later, but I'm sure every one of you knows that a 3-base sequence called a codon specifies a specific amino acid. At the very far end of this messenger RNA, we see codons for tryptophan. And these codons for tryptophan affect whether or not termination happens or no termination happens. Okay? These affect whether termination happens or no termination happens. So these, when there's plenty of tryptophan around, okay, cause termination to happen. And these, when there's no tryptophan around, cause no termination to happen. Now, why do I show you that? Well, the reason I show you that is it turns out that E. coli uses this mechanism for several operons for amino acids. Valine is also controlled in the same way. Leucine is controlled in the same way. Okay? So this is a mechanism for controlling operons related to amino acid metabolism. And that's as much as I'm going to say about the mechanism. Okay. Questions about that? You guys worn out? Are you ready for a song? All right. I wasn't going to do a song, but I'll do a song right here. How about that? This is to the tune of Frosty the Snowman. It's called Transcription. And you got to sing loud, right? Okay. Phosphodiesters are the bonds of RNA that support a ribopolymer made of G, C, U, and A. The RNA polymerase binds to a Tata box and copies from the template strand all along the way it walks. Initiation of transcription thus proceeds. From the closed to open complexing in the DNA it reads. The sigma factor gets released, its work is over fast. Polymerase can then advance after this step has been passed. In elongation, the polymerizing spree moves along the way in fits and starts synthesizing 5-2-3. The RNA is floppy and it dangles from one end. Oh, that's a most important thing for you to comprehend. Then termination finishes the RNAs, thanks to protein row or hairpin forms that release polymerase. So this describes transcription steps in 3-part harmonies. Here's hoping with this melody you can learn it all with ease. And that's the end. All right. See you guys Friday.